Okay, so good evening, everyone. Welcome. Happy New Year. It's great to be back. I'm really excited. This project has been going. This is a pre-COVID project. It's been going now for over a year. It's we really wanted students to run student-led seminars run by students for students to present what originally was supposed to be research on campus, and since COVID has been international. So it's great to be back. And today I'm really excited that we're having our second joint program, thanks to a Magistérien who's abroad. So thanks to interaction with Clarisse, who's going to present Fiona. Today we have two of my favorite female scientists. So Fiona I've known since I was a postdoc and continue to watch her wonderful career. And if you want to hear more about Fiona and her past, including her early days as a crocodile hunter, then go and listen to the Lonely Pipette podcast episode number 4, where you hear all about Fiona. And the other person is Claire, who's who's an amazing colleague who I've been working with and we set up together the Center for Epigenetics. So it's really exciting to have them and to see faces and names from Magistériens of the past. I can see actually all across Europe, and especially happy that Clarisse is going to present Fiona. Thanks everyone for coming. We want to keep this going, so please come back if you have any suggestions for speakers or people that you would like to hear, or people or postdoc or student groups in other cities that we can twin with. This would be fantastic. And I did promise you, and it's a great start, that 2021 is going to be the best scientific year that you've seen so far. And I think Claire and Fiona will prove what a great start. So thank you very much for coming. And Clarisse, over to you.
Hi everyone, thank you for joining. It's actually pretty great to be here now because, as Jonathan said, I'm a previous student of the Magistère, and now I'm here like one of the alumni presenting my boss for a talk. So that's actually pretty cool. So I'm really delighted to introduce Fiona. So Professor Fiona Watt obtained a first degree from Cambridge University and a Doctor of Philosophy degree in cell biology from the University of Oxford. She was a postdoc at the MIT, where she first began studying differentiation and tissue organization in mammalian epidermis. She established a first research group at the Kennedy Institute of Rheumatology and spent 20 years at the Cancer Research UK London Research Institute, which is now part of the Francis Crick Institute, a very famous institute here in London. She helped to establish the Cancer Research UK Cambridge Research Institute and the Wellcome Trust Centre for Stem Cell Research in Cambridge. And after that, she decided to move in 2012 to King's College London to found the Centre for Stem Cell and Regenerative Medicine. She's currently director of that center at King's College London, and she's the executive chair of the Medical Research Council in the UK. And I'm really impressed and pleased to say that she's the first woman to lead the MRC since its foundation in 1913. So Professor Fiona Watt is a fellow of the Royal Society and a fellow of the Academy of Medical Sciences, an honorary foreign member of the American Academy of Art and Science. And she was recognized for her work by many awards, including the American Society for Cell Biology, the Woman in Cell Biology Senior Awards, the Unitarian Society Medal, and she received as well the Amber Woman in Science Award. She is internationally recognized for her work on stem cell and their interaction with the niche in healthy and diseased skin. And today she's going to present her work, and the title of her presentation is Exploring Skin Cell Heterogeneity. So that's a great honor to have you today, and I just pass it over to you.
Fantastic, thank you, Clarisse. So I can share my screen now, right?
Yes.
And can you see the slides?
Yes.
Great. Well, it's lovely to be here, and I would like to give a very, very strong endorsement for the Lonely Pipette series. I was very doubtful when Jonathan contacted me. But what I do, I should say, is like on a Saturday morning when it's difficult to unwind, I just listen to one of those episodes and they're so amazing. For example, I discovered that that Robert Weinberg's father was a dentist, just as mine was. And in all the years I've known him, this was not revealed. So I would highly, highly recommend listening to this series, and congratulations, Jonathan. The other thing to say is that since lockdown, I've had all terrible experiences of giving online presentations that you can imagine. So I'm kind of shock proof. But if necessary, Clarice she's got my slides, I will phone and we'll take it from there. Anyway, without further ado, this slide shows you where I am sitting right now. So our labs are at this top floor of Guy's Hospital Tower, which is next to the Shard. And although it would be much nicer to be all together in Paris, at least London now is quiet, and it gives you a good sense of perspective when you think about landmarks like St. Paul's Cathedral over there and all of the different challenges that humanity has faced over many years. So anyway, I'm going to tell you about the work that we've been doing on single cell RNA sequencing in skin, human skin. But before that, I want to put the work in context by telling you about our interests in heterogeneity of the cells. I'm going to spend most of my time talking about our work on the epidermis, but then I'll give you a tiny flavor at the end of work that actually has been led by Clarice, looking at subsets of other cell types in the skin. So this is a very simple H&E stained section of adult human skin. It's a region where there are no hair follicles. It's just a very simple structure. And there are two layers, the outermost layer, the epidermis. This is a multi-layered epithelium, and the deepest layer of cells, the basal cells, are attached to a basement membrane. And the basement membrane sits on top of the underlying connective tissue called the dermis. And you can see that the outermost layers of the epidermis are cells that don't have any nucleus. They don't actually have any cytoplasmic organelles, but these cells are very important to protect our bodies from the environment. And as these cells are shed, they're replaced through the proliferation of cells in the basal layer down here. The journey from the basal layer to the outermost layers is about a month in most body sites. And because undifferentiated cells are giving rise to differentiated cells on a regular basis, this is one of the classic adult tissues which is known to be powered by stem cells. My interest in the epidermis came from my postdoc with this man here, the late Howard Green, and back in 1975 with his student Jim Reinwald, he reported that you could take a biopsy of normal human skin, disaggregate the cells, and then plate the epidermal cells at clonal density on the feeder layer of 3T3 cells. And when you did that, you'd get little colonies of cells forming, and they spontaneously formed multi-layered colonies. And over time, those individual colonies merged to give you a multi-layered sheet. Now, Howard had trained as a doctor, although he never practiced. And I believe that it was because of that medical training that he immediately realized the clinical potential of expanding human epidermis in culture. And only 6 years after that first report, he had shown that he could successfully graft the arm of a man who had suffered a burn injury and get permanent coverage of the epidermis using the person's own cells expanded in culture. So this is not only a classic tissue that is maintained by stem cells, but a classic stem cell therapy. Now, I told you that one of the things about this cell culture model is that the cells grow as colonies. And to the present day, we use colony forming ability of human epidermal cells as a quantitative readout of stem cell number. So this shows you several plates of cells that we've grown. We've taken the feeder cells away, we've stained the individual colonies of keratinocytes, and we count colonies. In this case, we wanted to compare different batches of serum and decide which was the best to buy. And in the early days, my strategy for looking for stem cell markers was to pick proteins that were on the surface of basal cells and use flow cytometry to see if I could enrich, when I enriched the populations, if I could enrich for the clonogenic cells. And starting off with high expression of integrin extracellular matrix receptors, using this and other strategies, we were able to identify a number of markers which we believe enrich for stem cells. You might think, and you'd be right to think, that growing colonies of cells in a dish is a very artificial assay, but it's really remarkable that if you are interested in skin diseases, there are a number of examples, such as this one here, where you can actually visualize clones of cells on living skin. In this case, the clones correspond to clones of cells with a genetic abnormality, but it shows how we can relate the behavior of the cells in the dish to what is happening in patients. Now, over the years, my lab and many other labs have found markers of the different layers of the epidermis, and we've characterized a number of the factors that regulate the movement of cells from the basal layer through the upper layers as they undergo this terminal differentiation process. But a few years ago, we really wanted to focus on one aspect of this process, and that was a process that we call commitment. So we know that we have stem cells in the basal layer of the epidermis, and we know that some cells leave the basal layer to undergo differentiation. But we didn't really have any markers that told us the identity of cells which had, if you like, made that decision to leave the basal layer. So to go looking for markers of commitment, we went back to a Howard Green study from 1977, which we had sort of elaborated on some years later. And essentially the observation is that if you take colonies of keratinocytes that you've been growing on a dish, disaggregate them, make them a single cell suspension, and you hold them in suspension, then they will undergo differentiation in a synchronous fashion. So at time 0, we enrich for the stem cells, which have high levels of beta-1 integrins. At 24 hours, we enrich for cells which have differentiated, and our favorite marker is this protein involucrin. And what we found was that at 4 hours, the cells still have integrins. They haven't yet started to express involucrin, but nevertheless, they have made the decision to differentiate because we can't block differentiation, for example, with extracellular matrix proteins. And just to bring this to the present day, there are several qPCR plots that you can see down here, where we're looking at mRNA levels for 2 stem cell markers, so an integrin and a transcription factor. Then we're looking at involucrin and another differentiation marker. And I just draw your attention to this time point 4, where we have commitment. And you can see that at this point, the stem cell markers haven't yet declined. So to find out what is going on at that 4-hour time point, Ajay Mishra, when he was in my lab, started a collaboration with Tony Lai and Angus Lamond, who are proteomic experts in Scotland. And so we did the experiment of putting cells in suspension, checking that the cells were differentiating by looking in our colony forming assay, and then carrying out a transcriptomic analysis, but also SILAC labeling the cells and carrying out proteomics. If we looked at time 0 or at 8 hours, there was a pretty good correlation between mRNA and protein. But if we looked at 4 hours, the commitment point, that relationship had broken down. And in particular, we found that this 4-hour time point was dominated by a large number of protein dephosphorylation events. And so this got us to start looking at protein phosphatases and how they were expressed in cultured epidermis. This work was published 3 years ago, so I'm not going to take you through all of the details. But if you look down here at these qPCR plots, I've already told you how the stem cell marker declines in suspension. And here are examples of 3 protein phosphatases where you can see their expression goes up at 4 hours and then declines after that. And to look at the functional significance of the protein phosphatases, we identified a total of 22 which were upregulated at 4 hours. And here we did knockdown experiments where we looked at the effect on colony formation. So in this spider web diagram, each blue circle represents 10% of the cells forming colonies. The red is the control. So in the scrambled control, about 20% of the cells that we've plated will form colonies. And then you can see with the knockdowns that knocking down some of the phosphatases gives us a very big increase in colony formation. So in this case, we could see that this transient upregulation of DUSP6 probably serves to drive cells out of the stem cell compartment, because if we knock down expression, we get more colony formation. We can look at where the protein phosphatases are expressed in the basal layer of human epidermis. And these look like carpets that you can see here. In fact, this is the basal layer of the epidermis stained as a hole mount. And up here, you can see the groups of cells with high levels of DUSP6, and they correlate with the cells enriched for stem cells, which have high levels of integrins. There's another phosphatase here, DUSP10, which shows more uniform expression. And here's an example of a phosphatase which shows patterned expression, but in the opposite pattern to the stem cell markers. So we found these phosphatases. We think they're important in culture, and we can see that they are indeed expressed in the epidermis in vivo. How do they regulate differentiation? Well, I told you that we know a lot about the differentiation process. And so when our collaborators looked at the top 15 dephosphorylated peptides in the data set, we found that 3 of them were ERK MAP kinases. And we know that decreased ERK signaling drives cells out of the stem cell compartment. And we also know that keratinocytes express a large number of AP1 transcription factors. Some of them, like these 2, MAF and MAFB, are induced with time in suspension. And when we knock down the phosphatases, we could reduce that induction. So we're pretty confident that this commitment event that we're looking at, the phosphatases are linked to ERK signaling and AP1 transcription factors. We knock down individual protein phosphatases, as I've said, but we noticed that when we knocked down expression of one phosphatase, it affected expression of the others. And this is a heat map showing you real levels of mRNA, and it's showing you what happens, what the relationship between the phosphatases. So an arrow shows you a positive impact on expression, and a T-bar shows you a negative impact. And just looking at this, you can see that there are a lot of positive interactions at 4 hours, far fewer when the cells have begun to differentiate. So we were able to give our data sets, the proteomic and the RNA data for the different time points. And in collaboration with Sarah Jane Dunn at Microsoft in Cambridge, she was able to make a Boolean network analysis, which was consistent with the concept that the stem cell state is stable, the differentiated state is stable, but this transient upregulation of the protein phosphatases acts as an unstable commitment switch during differentiation. More recently, we've been looking, rather than looking at bulk populations of cells, we've been looking at single cells. And this is the work of Toru Hiratsuka in the lab. And Toru had done his PhD with Mishi Matsuda in Kyoto, and so had a lot of expertise in FRET sensors, and in particular, using FRET sensors to look at ERK signaling in individual cells. And the particular sensor that Toru has been using is very useful because it reports in the nucleus. And what Toru was able to do was to introduce the FRET sensor into our cultured human epidermal cells and combine that with a reporter of differentiation. So you can see this is M-Cherry driven by the involucrin promoter. And what Toru noticed was if we looked at individual cells, this is 4 individual cells that Toru has monitored over a period of up to 16 hours, that we frequently saw that before the involucrin reporter was switched on, there was a period of fluctuating ERK activity. So you can see that in each case. The periodicity of the fluctuations was not constant, and the size of the fluctuations was not constant. But what we could say was that it seemed that fluctuating ERK activity preceded differentiation. And of course, the idea that we've got this unstable commitment switch that is impacting on ERK fitted very well with Toru's observations. Toru went on to see what effect 2 of our phosphatases had on ERK. And he did this using a doxycycline inducible vector in each case. And what he saw was that if he induced expression of DUSP6, there was no effect on mean ERK activity, but pulse levels went down. And conversely, if he looked at DUSP10, there was no effect on mean activity. Sorry, there was no effect on the pulses, but mean levels went down. So the hypothesis that we published a few months ago, based on the data that we have, is as follows. We believe a feature of a stem cell is that it has high levels of high stable ERK expression, and of course does not express involucrin. But when the cells differentiate, they have low levels of ERK and they switch on involucrin. And in the pulsatile state, if the cells are pulsing and they have high ERK levels, they can return to the stem cell state. But pulsatile low mean levels are indicative that the cell is committed to differentiate. So I'm going to summarize everything that I've said in this slide here. I told you how we were interested in that event when the cells leave the stem cell compartment, which we describe as commitment. I presented evidence that it's an unstable switch, which is regulated by protein phosphatases, which interact with one another and impact on ERK signaling. And I haven't got time to show you this, but in really beautiful experiments, Toru showed that pulsatile ERK activity is a feature of differentiating cells in living mouse epidermis. So at this point, I want to switch and say, what can we get out of this with the lens of a global initiative called the Human Cell Atlas? This was kicked off in October 2016 by Aviv Regev and Sarah Teichmann. And the idea is simply to map all of the cells in the human body with the vision of getting new insights into human physiology and disease mechanisms. And the Atlas has been embraced all over the world, and it's driving new approaches to integrating data, collaborating and sharing data. And I was really excited to be invited to go along to the kickoff meeting, because with my then postdoc, Kim Jensen, who's a professor in Copenhagen, I'd been very interested in whether we could use single cell gene expression profiling to find new stem cell markers. And I just wanted to share our early attempts at this. This was a paper we published in 2006. And in the work that Kim did, we could only be confident of differences in gene expression that were at least 7-fold. So we couldn't pick out our integrin markers using this approach. And we looked at a total of 300 basal cells. We found 9 stem cells expressing 2 of our markers. And that's a big labor, as you can imagine, to look at 9 cells. Luckily, it ended well because Kim found ELRIG1, which turned out to be an important regulator of the stem cell compartment in pretty much every epithelial cell that you can find. In fact, it's important in other cell types too. But you can imagine the idea that we could scale this up and collaborate was really great. So the 2 predictions we had were, first of all, that there are multiple populations of keratinocytes in the basal layer of the epidermis. And secondly, that the protein phosphatases are important here. And there are a number of different ways that you can analyze this. This is actually taken from a review which is talking about doing single cell analysis in the kidney, but the strategy is the same. You take your tissue, you disaggregate it in different ways, then you can make use of a number of different techniques to do single cell RNA sequencing. And then what you want to be able to do is to map the data onto, oops, I haven't died. It's the light gone off. You want to be able to map the data onto where the cells are in the tissue. And we've had a really great collaboration led by Muz Hanifa and Sarah Teichmann, and led at King's by Magnus Lynch, with Clarice really being incredibly important for this, as Christina, Daria and Bebe have as well. But I told you that Kim and I, 14 years ago, had managed to analyze a total of 314 cells. Look at the number that we've been able to analyze today. So we've been able to look at adult healthy skin, skin from patients with atopic dermatitis and psoriasis, and we've also had adult access to fetal skin. So this has been just really exciting to be able to analyze. And what I'm going to show you first is my former postdoc, Victor Negri, who extracted from that large data set information about the epidermis specifically. And of course, because we've studied it for so long, we can be very confident that the basal cells express keratin 5 and 14, the differentiating cells express other keratins and markers that we know about. And so using UMAP plotting, Victor has been able to confirm our prediction that not all basal cells are the same. In fact, in this analysis, he has found a total of 3 clusters, and our stem cell markers tend to lie in this population here that we call basal 2. We've got differentiated cells here, but what about these 2 populations that Victor's described as transition 1 and 2? Well, we were absolutely delighted when we went in and looked at transition 1 and 2 to see that our phosphatases DUSP6 and DUSP10 mapped here. And furthermore, if we go back to the original qPCR experiments in suspension, we knew that DUSP10 expression was elevated for longer than DUSP6. And that fits very well with the observation in the UMAP plots that the DUSP10 expression is distributed over more cells than DUSP6. Another thing that Victor has done is to look at the relative abundance of the different populations in healthy and diseased skin. And what he sees here is that not only is the size of the transition population reduced in healthy skin, but that's also true for the basal populations. And instead, we see an expansion of the early differentiated cells. Now, that in itself is not surprising because by histology, we've known for a long time that the spinous layers are expanded. But what's exciting is that it gives us new opportunities to come up with potential mechanisms by which the differentiation process is deregulated in the epidermis. So the human cell atlas data that supports the predictions that we had in culture, and it's giving us new ways to devise in vitro experiments because it's giving us very different ways of thinking about how the epidermis is regulated. Now I'm almost finished, but I wanted to end, as I said, with looking at other cell types in the skin. And this is taken from our collaboration with Maz and Sarah, and it just shows you the extraordinary richness of the different cell types that we can look at. We can not only look at what's happening in the epidermis, we can look at what's happening in the dermis, and we can look at different types of immune cells. And one of the cell populations that we've been interested in for a very long time is connective tissue, the fibroblasts of the dermis. And work which was really kicked off our interest in this came from studies that Ryan Driscoll initiated, where he was working in the mouse. And he realized that fibroblasts actually are multiple cell types. he was able to define several distinct populations of cells, and using lineage tracing, gene knockouts, and transplantation experiments, he showed that they had different functions. So our predictions from studies in the mouse were that there's more than one kind of fibroblast, that they have different functions, and actually they're characterized by differential expression of a number of different signaling genes. We wanted to test the function of the different fibroblast subsets without going back into the mouse. And in work that Christina Philippius has led, we used this classic technique for functional studies. So what we do is we take the epidermis off a piece of surgical waste skin, we take out all of the cells from the dermis, and then we rebuild the dermis with different populations of fibroblasts, put epidermal cells on top, and have a look at the quality of the epidermis. And we feed the cells in these microwells from below. So if we don't add keratinocytes, you can see the surface of the reconstituted skin is shiny, but if we plate keratinocytes on top, we make quite a nice epidermis. And using this assay, Christina, in collaborating with Daria and with Magnus, was able to confirm that in human, as in mouse skin, we could find different populations of fibroblasts with different functions. Just to illustrate that, this is collagen 6A5. It's a marker which is specifically expressed in the fibroblasts closest to the epidermis, as you can see down here. And here's a different marker, CD36, which is expressed in the lower layers of the dermis and also in the subcutaneous fat. And here are just two examples of skin reconstitution models. Here's a population of fibroblasts that are good at supporting epidermal reconstitution, and here's a population that are not very effective. So now using the Human Cell Atlas data, we've got a huge data set to analyze, and we've been able to confirm that there are indeed multiple fibroblast subpopulations. We found new markers which are important for our studies and candidate mediators of functional heterogeneity. Now, the last thing that I want to say is a question that is of great interest to me, and this is one that Clarice is leading on. What I would like to know is whether it's possible just to look at the skin and be able to predict from that, without taking a biopsy, that whether or not there are changes in the underlying cellular composition. And the approach that we've taken for these studies is to use a technique of non-invasive imaging called OCT, Optical Coherence Tomography. It's like an ultrasound probe that you run over the surface of the skin, and you can build up this really nice 3D impression of how the skin looks. You can analyze the blood vessels, as you can see in this network here. You can also quantitate the thickness of the epidermis. So in psoriasis, the epidermis is thicker than in healthy epidermis, and we can visualize that here. So Clarice has done a lot of analysis of the density of the blood vessels in different body sites, and these are just some examples from her unpublished studies. And you can see in this image here from forearm, the number of blood vessels and their branching is much less complex than if we look here at the lip or the forehead. So the question is, if we analyze skin from different body sites, can we work out what the cellular determinants are of that complexity of vascular signaling? We don't have the answer to that yet, but you can see how it becomes very interesting to ask these different questions. Which cells types might be important? What sorts of communication are important? So I just want to end by saying that the single cell RNA sequencing has been very useful because it's confirmed the existence of some of the cell populations that we had predicted existed. It's an important reality check for our experimental models. It's given us new ways to collaborate with others and share the data. And most importantly, of course, we believe that it's going to tell us new things about skin physiology and disease mechanisms. Last slide is just to thank my lab. This was taken at Christmas 2019. when we had no idea that we wouldn't be able to be less than 2 meters apart. But we've managed to collaborate very effectively and continue to work. So thank you, and I'm very happy to take questions. I'll stop sharing my screen.
Thank you very much, Fiona, for this really nice presentation and for really nicely showing all the work you did since the beginning of your career. That's really impressive. Even if I knew all about it, it's still like very impressive to hear about it. If you have any question, please feel free to write your question on the chat. I think Jonathan started with his question. So his question is, can you map the single cell population back into the skin morphology and organization?
Yes, Jonathan, we can do that. For some tissues, the emphasis, there's relatively little is known about the different cell types. We had the advantage in the skin that we already had some antibody markers. But what Clarice has done a lot of recently is using single cell RNA fish to map the cell populations. And Magnus has been developing computational tools which makes it easier for us to map where the cells are. So I think it's really important to know where we are. There are a lot of spatial mapping tools that are available, but so far we've done antibody mapping to say where are we, and now the RNA fish on top of that. We would like to do other techniques like whole mount labeling with where you render the skin completely translucent. So there are lots of interesting things we can do there.
Yes, absolutely. There is another question from Jonathan again on the ERK work. What sets up the ERK's pulses and regulates them in terms of intensity and frequency? So
we know that what sets them up is any of our known differentiation triggers. So for example, if we reduce integrin mediated adhesion, if we treat with a high level of EGF, which triggers differentiation. So in each case, the external trigger for differentiation sets the pulses up. We haven't seen any evidence that their intensity or frequency is controlled. We're not seeing that level of control. It's just the fact that the cells are in a pulsatile state.
Thank you. I think people are a bit shy here and maybe too impressed to ask any question. So if you have any question, you can even unmute yourself and ask. There is a question from Ahmed. Ahmed is actually a guy from my promotion in the master, so I'm really pleased to see that he's here connected. So his question is, could you perform velocity analysis on the single cell data? Did you get interested? Did you get interesting insights?
I don't think we've done that, Clarice, have we?
Sorry, I'm mute. No, we didn't. Yeah. But that's actually quite something interesting that we could do.
Yeah, that's a really good suggestion. And the other thing is that we don't have to do it. You can do it, because the data, an aspect of the Human Cell Atlas is open sharing of data. So one thing we did during lockdown was we became bioinformaticians, more or less. And so it's really great to analyze other people's data and then, and share that.
So there is another question. Yeah, that's true, actually. There is another question from Jonathan again. So it looks like that the differentiation is controlled by the phosphatase, but what about the kinases? And are they regulated, or is all the phosphorylation balance regulated by phosphatase alone?
Yeah, Jonathan, that's a really good point. First of all, I told you we found the phosphatases because of all the dephosphorylated peptides, But in fact, we found that there's a very, very tight transcriptional correlation between the levels of ERK, DASP6 and DASP10. So we found the dephosphorylated peptides, but there's transcriptional control as well. And the kinases are clearly important. There's old literature showing that if you inhibit PKC delta, you can block differentiation. We think you can keep cells in the stem cell compartment if you do that. And we believe that you need phosphorylation of P38 to trigger differentiation, and many of the phosphatases impact on P38. So I tried to tell a simple story, but there's so much more that can be done there.
Yes, so there is another question from Rec. So he said, amazing talk, thank you so much. My question is, in regards to the heterogeneity of fibroblast functions, could there be a regulation of cell differentiation commitment coming from the fibroblast secreted factor, or the opposite, lack of fibroblast support with factor when put in suspension?
Yeah, it's a really good question. So you can culture keratinocytes without any fibroblasts. And in fact, if you put fibroblasts in the dish with keratinocytes, they tend to compete. And that's why we use 3T3 cells as the feeder layer. But having said that, if you look, I think the best evidence is in mice. You can see very close communication between the epidermis and the dermis. For example, if you activate Wnt signaling in the epidermis, you get a kind of a rejuvenation of the dermis, the fibroblasts start to proliferate. And conversely, if you manipulate Wnt signaling in the dermis, you can provoke a very strong epidermal response. So I don't think that you need fibroblasts to control whether a stem cell will become committed. But I'm sure in vivo, all of these, the factors, the composition of the basement membrane, signals coming from fibroblasts, neighboring keratinocytes, and immune cells, all together, they are controlling the system, and individual stem cells have to integrate all of these different inputs.
So I think that's all. There is one question that just come up from Anna Jule. Have you looked at the cell interaction in your single cell data, in particular during development?
Very good question. We haven't done it. It could easily be done. We just, it's just, you can imagine the excitement if you've gone from having 314 cells to look at to more than 500,000. It's like, where do we start? So yeah, all of those things can be done. And I should say that Maz, Hanifa and Sarah Teichmann are mainly interested in the immunology. So their focus has been looking at how cell-cell interactions in the immune system change during development and also in disease. So that's all there to be analyzed for sure.
Yeah, and that's true that there is now really new pipeline that we can use to study cell interaction, that we should look at it. The only kind of problem we have, or I have at the moment, is under this big data set. My computer just gets crazy when I start doing some analysis. Yeah, I think it was the last question for your talk. Again, thank you very much, Fiona. It was really interesting, and I'm really pleased that you could present in front of all my colleagues from my master and also the new generation of the Magistère.
Well, thank you.
So thank you so much.